Good morning, everyone. Good morning. Welcome to your organic chemistry final. I hope you studied. This is not an anxiety dream. This is real. No, but welcome back to campus. You are in the right place. You are at the Fruitful Aging, What Fruit Flies Tell Us About Longevity and Health with Professor Mitch McVey. My name is Sean Devendorf. I'm the Executive Director of Alumni Engagement and Annual Giving, and I hope to be one of the first people to welcome you back for your reunion weekend. So welcome back. Hope you have a wonderful time on campus. And whether you visited last month or this is your first step back in 50 years, we hope you find it refreshingly familiar, but also with a few updates and additions that hopefully you'll get a chance to tour and look at. So it's a home that's been renovated and added to, but with very much that feeling that remains the same. In my role, I do have the distinct pleasure of getting to know faculty members and their incredible research and what they do and the impact they have on students and the research profile of the university and society. And you'll hear about that today with Mitch McVey, who's really embodiment of the Jumbo ethos. But before I introduce Mitch, just quick housekeeping, please silence your cell phones. We will have time for Q&A at the end, but I just ask that when we start the Q&A, you wait for a mic to be brought over to you. There are some people joining online today as well, and we are recording it. So now on to Professor McVey. Dr. McVey earned his BS in biochemistry at the University of Colorado at Boulder. After teaching high school for 2 years in the Rio Grande Valley with Teach for America, he moved to Boston to pursue his PhD at MIT, where he studied the role of genome instability in yeast aging. He continued researching DNA repair in his postdoc at UNC Chapel Hill, focusing on a more complex model organism, Drosophila, the fruit fly. He started his own lab at Tufts in 2005. He currently leads a team that is investigating what happens when cells make mistakes while fixing damaged DNA with a focus on how these errors relate to human disease and aging. The lab's research has been supported by the National Institutes of Health, the National Science Foundation, the American Cancer Society, the Elson Medical Foundation. Dr. McVey was named an AAS Fellow in 2023. He is the Director of Graduate Studies for Biology at Tufts and is co-director of the Tufts IRA-CDA postdoctoral training program. He is passionate about broadening participation in scientific careers and promoting scientific literacy. So please help me give a warm welcome to Professor Mitch McVey.
All right, good morning, everyone. Thank you for being willing to spend the first hour of your reunion weekend with me this morning. It's great to see all of you here. And if you're online, thanks for joining me as well. I'm just curious, how many of you ever took a class in this room? Anyone? Wow. How's it? These are new?
New to me.
All right. Excellent. This is the first time that I'm actually teaching in this classroom. I'm in the biology department, and so usually this room's reserved for chemistry folks. So it's really great to be here. And I want to do two things with you today. Number one, I want to give you a taste of what it's like to be in one of my classes. So I'm actually going to have you kind of go back in time and experience what it might be like to be an undergraduate in my biology of aging class. And then I'm going to talk a little bit about the science that the students in my lab do. I'm also going to try and tell you a story that we're hoping to get published in the next month or so. It's definitely one that made me scratch my head for many years while we've been doing this work with the undergraduates. And so I feel like sharing it with you this morning. So if at any time you feel like you have a burning question and you don't want to wait until the end, I am happy to take those in the middle, especially when we're talking about the science. So just kind of do this, and I'd be happy to answer questions along the way as well. All right, sounds good. So I'm going to set the stage for the talk today. When I teach my biology of aging course, we talk about drugs and supplements and interventions that can actually slow the intrinsic rate of aging or the intrinsic aging process in humans. And I have up here a list of 5 that in the media and in the popular press have been talked about in the past few years with regards to whether or not they might actually slow down aging. So I'm going to give you a little time here to look through these, and then I'm going to ask you to actually vote for which ones you think there's scientific evidence for. All right. We'll go in alphabetical order here. That's how I put them on the slide. So the first one is consuming fewer calories. Just raise your hand if you think that consuming fewer calories will slow the aging process in humans. I'm seeing about 2/3 of your hands going up. All right, you can put them down. Thank you very much. The answer to this is maybe. Okay, and the reason for this, it sounds like you're very well educated about this, by the way. The reason for this is there have been many studies that have been done in non-human organisms, including in monkeys, in fact. And the evidence is mostly positive. In most of the organisms, it does slow down the rate of aging. In monkeys, it's kind of mixed. It's unclear when they did the primate study whether or not that works. The reason we don't have hard and fast evidence in people that consuming fewer calories actually slows down aging is because it's so hard to do the study, right? Asking people to actually reduce the amount of food that you're consuming by about 20% for a couple years, which is what you would need to do this, not a lot of people can do it. And so in the studies that have been attempted with this, usually most people don't hit the target and they consume about 10 to 15% less of calories. Certainly their health span has been shown to improve. Whether or not it's actually extending their longevity and allowing them to live longer remains to be seen. All right, so that's the first one. How about drinking red wine? Show of hands if you think drinking red wine will make you live longer. Whoa. All right, I'm only seeing 2 or 3 hands going up here. More skeptical, yes? Nope. It does not. Yes. Oh.
We were in a lecture a couple of years ago.
Yeah.
They were talking about Alzheimer's.
Yes.
And the two things that they showed would help prevent that was green tea and red wine.
Yeah. Yeah. So there are certainly compounds, right? One of them in red wine is called resveratrol, and in green tea, there are also antioxidants. They certainly promote health span, and in the case of red wine, cardiovascular health, but there's no hard and fast evidence that in people it will slow the aging process if you drink the red wine. Doesn't mean that it's necessarily bad for you. We're just talking this morning about slowing the aging process. All right. Exercising. Show of hands here. I'll raise my hand here as well. All right. You can put your hands down. It does extend health span, which means if you exercise regularly, however many years you live, they will probably be healthier, right? And probably happier as well. Unfortunately, there's no evidence that exercising will slow down the intrinsic aging process. Certainly extrinsic, right? So it's going to decrease diseases of old age. But if you think it's going to add an extra 15 years to your life so that you can maybe break the world record, which is 122 years old, at the moment, there's no evidence for that, for exercising. All right. High doses of vitamins. I was told by my mom when I was 5 years old that I need to take my vitamins, right? Does that extend lifespan? Any hands? Not a one. It's controversial. Okay, so probably they don't extend lifespan, but as to whether or not it's a good idea to take vitamins, and we talk about this in my class, it really depends, right? So certainly if you have a vitamin deficiency, yes, that makes sense. But it turns out, and I'm going to try to lay the groundwork for the science that I'm going to talk about later, it turns out that for most of us, we get enough antioxidants and vitamins in our normal diets. And in fact, if we overload our systems with extra antioxidants, that can somewhat kind of undermine our body's own defenses that we have evolved over millions of years to deal with some of the things that vitamins would take care of. So that's why I put this as controversial. And you'll see how this ties into what we're doing in the lab in a few minutes. Okay, and last one, human growth hormone supplements. No hands. Definitely not, right? In fact, as we talk about in class, it turns out that if you have animals take growth hormone supplements, it will actually hasten the aging. They'll get bigger, they'll get stronger, but there seems to be this yin-yang, and by growing faster and growing stronger, that might actually decrease lifespan. So it might actually speed up the intrinsic aging process. All right, I'm going to give you an A, not an A+, but I'll give you an A today. Very nice job in terms of this quiz. So this is actually what I do in the first day of class in my biology of aging, just to set the stage for the students. And then we spend the semester talking about the science behind all of these things. The final project that the students do, and this is a class of about 50 students, is they work in groups to make YouTube videos. And these YouTube videos debunk some of the myths that are out there about anti-aging therapies and about longevity supplements. And so if you're interested at any point today or in the future, you can just go to YouTube, go at Biology of Aging Tufts, and you can see we've got more than 30 videos up there right now with the students talking about all sorts of things, including, for example, red wine, caloric restriction, green tea, curcumin, all sorts of things that you might be curious about. And I promise you that what they say in these YouTube videos is actually accurate because I make sure that they're going into the scientific literature and they're actually reading what's been published and they're critically evaluating it. So kind of a fun resource. Encourage you to check it out if you want to. Okay. So one of the other things that we talk about in the class, in addition to these anti-aging interventions, is, well, what actually happens to us as we get older, as we age? And there have been many, many, many biogerontologists who have studied this over the past probably 100 or more years. And it's starting to coalesce on that there are some hallmarks of aging that you can see up here on this slide. Okay, so for example, I don't know if there's, I prefer doing this. This purple box right here, stem cell exhaustion, right? As we're aging, we need to replenish important cell populations in our body, in our guts. And as we get older, those cells start to get tired. They have trouble making copies of themselves, and so that's called stem cell exhaustion. Sometimes our mitochondria, which you may recall, those are the powerhouses of the cell. They provide energy to our cells. Sometimes they get tired as well. They start to decline. What I'm actually going to focus on today of all of these hallmarks is the one that's the arrow's pointing to here, which is called genome instability. And that's just a fancy way of saying that as we get older, the blueprint of life that dictates everything that goes on within our bodies, which is DNA, it can get damaged, it can get tired, it can get worn out. And in fact, what needs to happen in order to promote long life is that DNA needs to get repaired and it needs to be maintained in as pristine a form as possible. Okay. This theory has waxed and waned over years. Right now, it appears to be waxing, actually, for several reasons. One is because, as I'm sure many of you are aware, genome instability, in addition to being potentially a cause of aging, is also directly related to cancer. And so this is Jan Vij, who is one of the pioneers in this field. One of the things that he has kind of postulated is that over time, as our tissues age, there will be things that happen to our DNA, mutations, for example, that change the genetic code, or that where the DNA starts to fray at its ends. This will cause a functional decline and could lead to cancer, or it could lead to cells actually dropping out and our tissues having trouble kind of repopulating themselves. Okay, so that's the idea behind this genome instability as a potential cause of aging. Now, there have been some high-profile papers in the scientific journals in the past few years suggesting that there's a lot to this theory. And it's kind of fun. I'm going to share with you some of the stories that have been published. The first one came out over a decade ago, and it suggested that elephants, which are all near and dear to our hearts here at Tufts, that they actually have many, many copies of a special gene called P53. P53 is sometimes called the guardian of the genome. It's mutated in many, many human cancers, and it looks like elephants have so many copies of this gene that it keeps their DNA in a very pristine form. And elephants live quite a long time. This could be one of the reasons for it. A second example, also interestingly enough, a very large mammal, is the bowhead whale. And it was published just last year that there's a particular protein in these whales. It's called cold-induced RNA binding protein, or CIRBP. Bowhead whales have evolved this special gene that's important for the repair of their DNA. And I don't know if you know or not, but bowhead whales live a long time as well. It can be up to a century in some cases, right? And it's interesting to think that maybe this improved DNA repair could be responsible for some of that. And then finally, it's always fun when I introduce this organism in class. The students either love it or hate it. I think it's kind of cute. It's called the naked mole rat. Naked mole rats, believe it or not, they tend to live in tunnels underground. They live extraordinarily long for their size, right? So they're only maybe 3 or 4 times bigger than a mouse, but they live 10 times as long as a laboratory mouse. And so there's a woman named Vera Gorbanova who has been studying naked mole rats at the University of Rochester for many, many years. What she published last year is that there's a special kind of variant of a gene that's called cGAS, and cGAS is important for DNA repair. It's found in the naked mole rats. when she takes that variant of that gene and puts it into other organisms, including fruit flies, they live longer. Which is interesting, right? Certainly, I'm not suggesting that we should take this variant gene and put it into people. That's not ethical. But just being able to realize that a single genetic change could increase lifespan by 2, 3, 4-fold, right? That suggests there's some potential for interventions, but potentially in people as well. Okay, so all of these animals live a long time. We talk about these in the biology of aging course, and it all seems to be kind of related to DNA repair. What I want to talk about today is how we have been studying this genome stability and DNA repair in our favorite organism. My lab is about 1.5 miles that way, close to the Mystic River. Tufts has actually rented out space in an old warehouse that's called 200 Boston Avenue. And on the 4th floor of this warehouse, you will see, I think there's about 12 labs from biology that are busy all the time with undergraduates, with graduate students trying to figure out all of these new things. And so we're in the corner of the 4th floor, and we work with this not to scale fruit fly right here. They're actually much smaller than that, thank God, right? And the reason that we've chosen to do the research with this model organism is what you see up here in blue, right? So it's a multicellular organism. You heard that I originally started my studies when I was a graduate student studying yeast, which is a single-celled organism, right? Really good for bread, really good for beer. You can actually study aging in yeast too. But when I finished graduate school, I said, all right, it's probably going to be more complicated in people. And if we really want to know what's causing aging in people, we probably need to study something that's got more than one cell. Fruit flies have a lot of cells, right? They have probably hundreds of thousands of cells. And nicely, they only live about 40 to 80 days. So you can do experiments fairly rapidly. But they do have a lifespan. And it can also be altered, right? So you can make them live shorter, you can make them live longer. The rate of aging, as you can see up there, then is modifiable. You can study aging in different tissues. What we're going to focus on today is actually the gut of these fruit flies and how that plays into aging. And I've been interested in genome stability for several decades now. Over time, with the help of students and postdocs and undergraduates, I've been able to accumulate a collection of DNA repair mutants that could have relevance to aging. And so you're going to see some of those today. Welcome. All right. So this was actually, and I'm going to show you some primary data now, but I'll make sure that I walk you through it here. These are survival curves from fruit flies that we raised in the lab about, I guess, almost 20 years ago now. And what you can see here is that we're looking at female and male fruit flies. And these graphs show days on the x-axis, so 0 to 100. That's about the maximum lifespan of a fruit fly is 100 days. And then the percent of the flies that are still alive on the y-axis. Okay. And you can see, and this would be true for any organism that ages, that over time, the survival rate starts to dip. What you're looking at here are 3 curves in females and males. The first curve in blue are WT, which are known as wild type flies. They're just normal flies, the flies that you could even find, for example, buzzing around your bananas in your kitchen. And their mean lifespan is about, I guess, 63 days for females, about 65 days for males. If you get rid of 2 different genes that are important for DNA repair, the names aren't important, okay, but you can see they're in green and magenta here. Look at what happens to the lifespan. Decreases, right? And not only the average lifespan, but also their maximum lifespan. So when we published these data, I guess about 20 years ago, one of the things we said was, okay, it looks like DNA repair is important for the health and for the lifespan of these fruit flies, and we wanted to know why. One of the nice things about fruit flies is if you have good eyes, and my eyes are not so good anymore, but the eyes of my students are good, they can use a microscope to dissect all the tiny little tissues out of the fruit flies. And so they did this, and we collaborated with someone at Tufts Medical School who's very good at histology, at staining. And what we did with him is we looked for tumors within the flies. What you're seeing here up at the top, those are basically sections of the fly gut that have been sliced right through the fly gut. So it's almost like you're looking down it. And let's see here. Yeah, this will be useful. So here's the outside of the gut. Here's the lumen. Here's the inside of the gut. This is where the food would normally be traveling through when it's getting digested. In normal wild type flies, what you see is that the gut is basically young looking. But if you get rid of a DNA repair gene, in this case, this is a gene called Bloom. It's mutated in a human disease called Bloom syndrome. See this mass of cells right here labeled with a T? That stands for tumor. That's actually a tumor that has come within the guts. It's grown within the guts of these flies, probably because they're missing this important DNA repair gene. You can also look at the germline tissues of the flies. So here from males, we're looking at testes. This is a normal testis in a normal fly that's getting fairly old. Here's a tumor found in the testis of this fly. Here's a normal ovary that you would see in a female fly. Okay. Right here, this is basically all of a tumor within that ovary. So just like people, fruit flies can get something that's very similar to cancer. And we asked the question of whether or not this changes as the flies get older, and in fact it does. And it's worse if you don't have DNA repair genes or if they're mutated. So you're looking at a very large bar graph here. We're looking at flies that were raised to either 35 days old, 50 days old, or 60 days old. We've separated them into males and females. And we're looking basically in blue at normal flies and in purple at flies that are missing a DNA repair gene. And what you can see is that as these flies age, there are more tumors, especially in the flies that are missing the DNA repair gene. So this was our first foray into aging in the fly lab. This was done in collaboration with Jan Vij, who's in New York, but also some undergraduates in the lab. And our conclusion from this was that DNA repair is important to prevent aging in the flies, and it looks like it has something to do with cancer in this case, which is really interesting because for people, right, cancer kills many. We'd like to prevent that. And so if we can learn some things in the fruit flies with how to prevent that, we could translate that into people as well. That's our hope for what we're trying to do here. Okay. So here's our take-home message. It's nice to be able to study this in fruit flies because some of the DNA repair processes that slow aging and promote the health span in these model organisms, they seem to operate in people as well. And at this point, we got interested in some other things in the lab. And for many, many years, we didn't go back to this until about, I guess it was 7 or 8 years ago. So this is 10 years after we published that study. I was at a barbecue, which was really fun. And I just ended up talking with a Tufts professor. And as these things often happen, we struck up a collaboration with something that he had been doing in his lab with mice. And that's the story that I want to tell you today. This is not yet published, so you're actually some of the first people in the world, besides those who have done the study, to be able to find out what it is that we've been up to. So I hope you enjoy this. All right. First and foremost, science is done by people. These are all of the people who have been involved in this scientific study. So the gentleman on your left, top left, that's Jim Crott. He was the person that I talked to at the barbecue. He's my collaborator. He used to be at the nutrition school that was associated with Tufts. He has since moved to Boston University, and he's got a lab that he's running there. All of these other individuals that you see here, these are undergraduates who have been through my lab in the past, I guess, 8 years or so. Interestingly, this project has not been touched by any graduate students, by any postdocs. It's only been undergraduate students who have worked on it. And I just think it's really fun, and it's really a testament to the quality of the students here at Tufts that they can do this high quality science, right, and hopefully have this published really soon. So I'll be highlighting them as we go along in terms of who has done what. Okay, so let me introduce you to the protagonist of this story. It's a bacteria. The bacteria is called Parabacteroides distostonus. For the rest of the talk, I'm going to call it P-dist because it's a lot easier to say. And Jim Crott, when I met him at the barbecue, told me that he had been feeding this bacteria to mice, and he had shown that in a model of colon cancer in mice, it actually attenuated the cancers. The mice got fewer tumors, fewer colon cancer tumors, and the tumors developed more slowly. Jim's hypothesis was that this bacterium has some sort of an anti-inflammatory property. So it's preventing something that aging scientists call inflammaging. Okay, and it's basically tamping down inflammation so that we don't end up with cancer and other things that are associated with that. So these are data from a paper that he published 6 years ago, in which he was looking at a mouse model of cancer. Here's tumor incidence. This is the control. Okay, remember, these mice have been bred so that they normally develop cancers. 77% of the mice developed cancers here. When Jim fed the mice the p-dist either early in their life or late in their life, in both cases, what he saw was a decrease in the number of tumors that developed and in the number of tumors per mouse. Didn't seem to have effect on the size. So once the tumors developed, they grew fairly quickly, but he was reducing the number of tumors just by feeding them this bacteria. And the crazy thing is, he had already killed the bacteria by freeze drying it. So the bacteria weren't reproducing within the colons of the mice. It was just something inherent in the bacteria, probably an anti-inflammatory property, that was decreasing the rate of colon cancer in the mice. That's cool. Yes?
Why did he choose to kill the bacteria?
That's a good question. So these are bacteria that normally exist. Yes. So the question is, why did he choose to kill the bacteria? And the answer is because he did feed them the live bacteria first, but he didn't want to go into a probiotic mechanism. He didn't want to actually have to have the bacteria be alive. So he just kind of tried it to see if he could kill them by freeze drying them, and it still worked. Interesting, right? Definitely. But that was fortuitous for us, because when we met at the barbecue, he said, well, hey, you should try this in fruit flies, because he knew that fruit flies could get cancer. And he said, and if it's an anti-inflammatory bacterium, maybe it'll actually do something good for their rate of aging as well. So what we did, and this gets to your question, right? Why were they heat killed? These bacteria can't survive in the guts of fruit flies, mainly because they can only live in regions of the gut where there's no oxygen. And fruit fly guts have oxygen throughout them, unlike mice and people. So the bacteria wouldn't be able to live in the fruit flies, but we could just feed them the freeze-dried variety. Very nice, right? Just go down to the store and get some freeze-dried p-dist. And so the way we do these experiments, when we grow these fruit flies, we raise them in plastic vials. The food is on the bottom of the vial. We can just drip the freeze-dried p-dist on top of the food, and the flies, because they need food, they'll eat the bacteria along with the rest of their food. Okay, so that's what you're seeing here. Basically, on the left, we have the control flies that are not getting any p-dist, and on the right, we have the flies that are getting p-dist. Good so far? Okay. All right. This was done by 2 undergraduates, Neha and Sarah. And what you're seeing here are survival curves again. I know there's a lot of messy things going on here. Let me give you the punchline. Okay. These are just different strains of fruit flies, males on the top, females on the bottom. The control fruit flies are the dotted blue lines. The flies that were fed the p-dist are the solid red lines. In all of these cases, we're showing you the median lifespan, and these percentages show the increase in lifespan by feeding the flies the p-dist. So 21%, 42%, 17%, 43, 28. In most of the experiments, we're extending the lifespan of these flies by 25 to 40%. That's a lot of years for people, right? If this were people. For the flies, you can see, right? They're living quite a long time. By the way, yes?
Male versus female in any of these flies?
Yeah. So the question is, why are we looking at male versus female? And the reason is that aging processes don't seem to operate the same in males versus females in many animals and also people. So it's really important when you're doing research, right? If you're studying an organism that has 2 sexes, that you're studying the process in both of those sexes. That's why we do that. Thanks for the question. Yeah.
Tease out whether it's the anti-inflammatory or something else.
The question is, can we tease out whether it's the anti-inflammatory properties of the bacteria or something else? We'll get there. Absolutely. That's what we've been doing for the past 8 years. That's what we've been trying to do. All right. So this is great. By the way, I didn't think this was going to work. When Jim told me this at the barbecue, I said, okay, I'll try it, but there's no way this will work. It worked. So now we got a project, right? And so we took this actually to, there was a funding mechanism at Tufts called the Tufts Healthy Aging Project. They gave us some seed money to do this project. we got some data like these, and then we applied to the National Institute for Aging, and Jim got a grant from them. So that's been allowing us to do this work. All from undergraduate research. Okay. We also wanted to know, could this make the flies not only live longer, but make them live healthier? Because it doesn't help if we're extending their lifespan if they're sick, right? So the way you study that in flies is you do something that's called a negative geotaxis assay. That's just a fancy way of you're asking, can they climb? Because as flies age, their ability to climb against the force of gravity actually decreases. Their muscles get weaker. Neurologically, they're not doing as well. So as they get older, if they can climb higher and faster, that means that you're improving their health span as well. All right, so here we go. Control on the left, pedist on the right. Those are the fruit flies in actual size right here. In a second, you're going to see me, not me, one of the undergraduates, tap them down to the bottom of the vial, and then they're going to start climbing. See if you can tell a difference. So the control, those are the ones without the pedist, without the bacterium, right? They're looking a little slow. This is about 50 days now of lifespan. The ones on the right, do they look faster? They seem better, right? They seem like they're doing better. Only difference here is that we fed them this freeze-dried bacteria. So this is one metric. We measured some others as well, but it looks like, right, not only are they living longer, they're living healthier. They're able to enjoy their lives more. All right. And we quantified this. This was Sarah's work again. Control in blue, pedist in red. The higher up you are, that means the flies are climbing farther in the vials as they get older. And you can see as they get older in both the control and the pedist treated, right? It's tougher to climb, but the decline is slower when we feed them the pedist. Okay. To your question, what are some of the mechanisms, right, that might be operating here? Why are the flies living longer? So we entertained 4 hypotheses. The first goes back to the very first question I asked you today, which has to do with caloric restriction. Maybe the pedist just tastes bitter or tastes bad, and maybe the flies are eating less food. And if they're eating less food, they might live longer, right? That's been shown by many other labs. So an undergraduate named Grace, who is graduating on Sunday, congratulations to her, she did an assay called the cafe assay, which is basically where you put the flies in a vial, you put a little capillary tube through the plug there, and then you fill the capillary tube with either sugar water or sugar water plus the pedist bacteria. And you ask, how much do they eat? Here are the results over here. NS is not significant. There's really no difference. So we can eliminate this first possible mechanism. It's not that the flies are eating less. This is not caloric restriction, which was good for us because otherwise that's kind of a trivial explanation for what's going on here. All right. Hypothesis 2. This gets to your question, Louis, is that right? Yeah. Which is, why did we heat kill them, right? Maybe what the pedist is doing is it's altering the microbiome of the flies. Maybe they have different bacteria now living in their guts, and maybe that's allowing them to live longer. So for this one, we collaborated with a scientist who's at the medical school. His name is Michael Malamy. What you're seeing on the left here is just the gut of a fly. Here's the gut of a mouse. These are the microbiota composition. These are just ways that you can study the different bacteria that are living in a gut. Oftentimes you see it as a pie chart like this. Here's the conclusion from our studies with Michael Malamy. We found that the pedist could not colonize the Drosophila gut, which we expected. What we haven't done yet is we haven't done actual microbiome experiments to see whether or not there's a difference in the bacteria in their guts. So this is still an open question, and I can't tell you whether or not their microbiome has changed. We'll need to do that, most definitely. Third, maybe these cells that are found in the guts of the flies, maybe it's changing the populations. Okay, so what you're looking at here is a cartoon of the inner lining of a fly gut. There are 3 different types of cells. Just like in our guts, we have lots of different types of cells. In flies, they have these things that look like they kind of have a flat top haircut on the top. Those are called enterocytes. Those are the ones that are actually going to be responsible for digesting the food of the flies. The one that you see that's called ISC, that stands for intestinal stem cell. Those are the cells that will divide whenever the gut gets damaged. Okay, those are really important because oftentimes when we're eating things we shouldn't be eating, right, it can damage the cells within our gut and we need to replace them. That's done with those intestinal stem cells. And so in flies, as they get older, stem cell hyperplasia, just a fancy word that basically means you get more stem cells dividing more rapidly. Okay, it's going really fast. That's a common thing in older flies. So Laura Kusaku, another undergraduate who is currently on a Fulbright fellowship in Albania, she dissected out the guts. Going to be hard to see these, but that's okay. She dissected out the guts of the flies, and then she used molecular biology techniques with a technique called immunofluorescence to look and see which cells were dividing within the guts. So any place that you see in these pictures, little green dots, those represent cells that are dividing. Okay, these are the stem cells. And when she took these pictures and painstakingly quantified them, took many hours at the computer to do this, what she saw is that in the young and the old flies that were not fed the pedist, there weren't a lot of cells that were dividing. But in the flies that were old and that were fed the pedist bacterium, large increase. Okay, so we scratched our heads a little bit here because I just told you that as flies age, you get more cell division, right? That's a normal thing with aging. These pedist flies are supposed to be living longer. In fact, they are living longer. But yet, by this metric, there's more cell division. That's the opposite of what we would have expected. And so there's a lot of head scratching that went on for a couple of years, right, in the lab for this. And that made us think about something that you may or may not have heard about, which is called gut barrier integrity. As many animals get older, as they're aging, the gut becomes leaky, which means that the things that are supposed to stay inside of the gut, they actually leak out into other parts of the intestinal tract and into the body. And that's a bad thing. That can cause the immune system to mount an inflammatory response. And so you could have inflammatory processes that lead to aging. So we said, all right, maybe pedist, the bacterium, is improving this barrier. It's preventing things from getting out of the gut, and therefore it's preventing the inflammation and the aging. The way you test this is using an assay that's called the Smurf assay. How many of you remember Smurfs from your childhood? I certainly do, right? Smurfs are what color? They're blue, all right? So the reason this is called the Smurf assay is you just feed the flies a blue dye. It's non-toxic. And because flies are a little bit see-through, you can actually see the blue dye when it gets into their intestines. And just through a microscope, you can see, is the blue dye staying in the intestines, like right here, or is it spreading throughout the fly? And if it spreads throughout the fly, that means the intestine is leaky, the gut barrier is defective, and so that blue dye is leaking all over the place, and you have a Smurf fly. All right, so undergraduate student who's currently in medical school at Harvard, her name is Luana Machado, she did this Smurf assay. In blue, you can see the proportions of Smurfs. Okay, remember, these are the ones that have leaky guts in the normal control flies and then the pedist flies. You can clearly see here, right, that if you feed the flies pedist, fewer Smurfs, you still get them, but fewer of them as they get older. So yes, it looks like this bacteria is promoting the integrity of the gut barrier, and it's probably preventing inflammation in that way. Okay, good so far? I think I've got about 5 minutes left here, if that's okay. All right. Okay, I'm going to skip this. All right, last piece of this puzzle. For the moment, all we've been doing is we've been dissecting out the tissues and we've been using some clever assays, but we haven't looked molecularly at what's actually happening. And for a molecular biologist such as myself, one of the key things to know here is how is the pedist changing the way that genes are being expressed? Okay, so our DNA is made up of genes. If you have biology background, you may remember that DNA gets turned into RNA, gets turned into protein, right? And so gene expression just means how good is the cell at turning the DNA into the proteins. To answer this question, we used a molecular biology technique that's called RNA sequencing. Sometimes it's called RNA-seq. It's a really cool thing that you can do where you can just take all of the RNA out of the guts of the flies that either did or didn't have the pedist. And using next generation sequencing technologies, you can basically count the number of DNA copies that are getting turned into RNA and protein. Okay. This was done by these three undergraduates in the lab, Ben, Grace, and Jill, and then Vicki Doe, who's now at Dana-Farber. She's a bioinformatician. She's done all of our bioinformatics analysis on this. She does all the computer stuff that I can't do. All right, so here you go. These are typical things that you would see in a study like this. I'm just going to tell you what they mean. Basically, any dot that you see in red there, that means that there is a significant difference in the expression of that particular gene in the p-dist compared to the control. Any dots over here on the right, so in this quadrant right here, these are genes that you make more copies of the RNA. Any genes on the left, these are ones that you make fewer copies of the RNA. And this is comparing p-dist to not p-dist. So all of these genes over here, all these red dots, could give us clues as to what's the mechanism by which p-dist is extending the lifespan. So Vicki took these data. She did her magic with the computer. She kind of classified all these different genes into categories. And what she found here is that p-dist, that bacteria, will turn on the expression. It'll cause more of the DNA to get turned into RNA and protein for cell division and DNA repair genes. Now think about what I told you at the beginning of the talk today, right? It looks like there's a lot of evidence for DNA repair being important for the prevention of aging. Here we have a bacteria in freeze-dried form that if we feed it to the flies, it is somehow turning on these DNA repair genes. How is it doing that? That's the final question, right? Well, I don't have the answer for you this morning. We're still trying to get there. But we do have a working model. Okay, so I'm going to float the model to you and feel free to shoot it down. I can take it, right? Scientists can handle this. All right, so here's the idea. Here's our p-dist bacteria. What we think it's doing is when it's going through the guts of the flies are recognizing this as foreign. They're saying this isn't supposed to be here. And they're mounting an immune response. Probably an innate immune response, which just means it's not going to use antibodies and all of the things that we often use to get rid of infections. Flies don't have antibodies. They're just using proteins that recognize foreign substances. And when they recognize this foreign bacteria, they say, all right, we got a problem here. And they mount something that's going to cause oxidative damage. Oxidative damage, that's a fancy way of saying we're basically going to take byproducts of oxygen and we're going to upregulate stress responses with those. In this case, the oxidative damage may be a good thing. It's not always. This immune response, we think, is now causing those cell division and those DNA repair genes to get turned on, like I showed you in the previous slide. And that's probably a good thing, right? Because if this were an actual infection, the fly needs to be able to clear it. But it's not an infection. We've tricked them because the bacteria are dead. They're freeze-dried, right? And yet the flies are mounting this response. And what we think is happening, this is my hypothesis, I don't have proof for this right now, is that this response continues. And it's basically having an effect that's known as hormesis. There's a popular song that you may have heard of that says, what doesn't kill you makes you stronger. That's what hormesis is, right? Stress that you might have earlier in life actually causes your body to ramp up its defense systems. And if they stay on at the proper level, they can actually slow down the pace of aging because your body's better able to deal with things that are going to come your way later. And so we think here what's happening is because those DNA repair genes get turned on, that's causing the gut barrier to stay intact, okay, so fewer Smurf flies, and we think it's also slowing the rate of aging. That is a hypothesis. I can't say that's what's happening, but that's what we do as scientists, right? As we make hypotheses. And now I'm going to work for the next 15 years to test this. So come back in 15 years. So this is what we're going to be trying to do, and I'm sure I'll be doing this with other undergraduates. We're trying to ask whether or not it is the innate immune system that's being activated by the p-dist, and is it causing DNA damage? I think the answer is going to be yes, but we'll see. Does it have any effect on the tumors that we looked at earlier in the talk? Are there fewer tumors in the flies? What component is responsible for this? We don't actually know what part of the p-dist is doing this. And then ultimately, we probably won't answer this in our lab, but I hope someone will. Could PDIST actually extend mammalian lifespan? Could it actually be useful for people? I don't know. It's going to be fun to find out. Definitely. All right. I'm done. You've already asked some good questions. I would love to hear more of them. So I think we're going to take time now for question and answer period. Thank you for your attention. They're going to bring some microphones to you. And then as you get the microphone, feel free to ask your question and we'll go. I think I caught him off guard. So we have one here and one there. Yeah. Yes.
Thank you. Very, very nice talk. Is there anything strikingly different between PDIST and your garden variety bacteria that we don't know?
That's one of the things we're trying to find out, right? So the question is there what's different about the PDIST compared to any other bacteria? So one of the things we wondered is, could we just feed freeze-dried any bacteria to the flies? Would that improve them? And so we took some E. coli, which is a garden variety bacteria, right, that you find in lots of places. We freeze-dried it. We fed it to the flies. It actually shortened their lifespan. So there's something special about the PDIST, and Jim is working on this. I'm not sure if he's identified the protein or the molecule yet, but yes, there's something special about the PDIST that's going to be important for this. There's a question here. People are reuniting as we speak.
The funding cuts from this administration,
Yeah.
how is that affecting your research?
Thanks for that question. Yeah. It's affected our research. It's affected a lot of people's research. So what we have found is that we also get some of our funding from a different institute at the NIH. It's called the National Institute of General Medical Sciences, NIGMS. Because of the funding cuts and because there are fewer people at the NIH to process grants, we actually waited for 1.5 years for a grant that I submitted in January of 2025. We just heard 3 days ago that we had gotten it. Phew. Thank you. But it hurt, right? I was afraid I was going to have to lay off some people because it took so long. I think we were one of the lucky labs, to be honest, because we ultimately did get the funding. It's hurting a lot. And basic science like this, people aren't going to be able to do it if this continues. Thanks for the question. Yeah. Yes.
Thank you. Fabulous talk. I have so many questions.
Of course.
I'm going to go with you're very, very generous in crediting undergrads for their part in the research.
Yeah.
2 parts to that. One is, realistically, how much of the brain power thinking through what experiments they're doing came from them versus from you?
Yep.
And second is, how competitive is it for undergraduates to get internships with you or lab experience with you? Are you getting a ton of applications, or are you having trouble finding undergrads to do the work?
Those are 2 great questions. So I'll deal with the first one, which has to do with, yeah, I'll repeat it. So the first question is, what's the intellectual contribution of the undergraduates to the research? Usually when an undergraduate first comes into the lab, they're new to it, right? And so the whole point is to involve them in the research, but I'm usually driving it for the first semester or 2. Some of the women that you saw, whose pictures you saw up there, they were in the lab for 3-4 years. By the time they were seniors, and several of these students were doing senior honors theses on this, we would sit down, right? And we were spitballing ideas back and forth. They're intellectually engaged. They're doing it. Not at the start, but by the end, absolutely. They're like graduate students. In terms of the second one, the other question was, how hard is it to get a position in my lab and in other labs? Right now, it's really tough. There's much more demand from the undergraduate population on this campus than we have spaces in the lab. There's the undergraduate chair in our department, whose name is Ben Wolf. He has organized a way so that students can apply equitably for lab positions, right? Because there's often a hidden curriculum. Some students know how to get lab positions, some students don't. So now it's been evened out, so everyone applies through a central kind of portal. And he's also involved a lot of labs in biomedical engineering and chemistry and downtown on the medical school so that students can get these opportunities. So there's still a great need, absolutely. And we love it when people are able to fund undergraduate positions. That's really helpful, but I think it's getting better. Thanks. Yes.
Hi. So I think it's generally understood, like in human populations that women tend to outlive men.
Yes.
But in your lab results, it seemed to show that the male fruit flies outlive the female fruit flies.
Yeah.
Is there an explanation for that, or is there any kind of correlation between human life and fruit fly life?
Yeah, good question. So the question has to do with, in humans, usually females on average live longer than males. In fruit flies, the males actually live longer than the females, at least according to these data. So what you saw was kind of a consequence of something in the lab, which is that when female fruit flies mate, the energy that they put into the production of their eggs actually shortens their lifespan. And so what you were seeing there is the females, they had mated, right? And so the energy they put into their egg production caused them to live for a shorter period of time. If in fact you don't allow the females to mate and they're not producing all of the eggs, they live longer than the males. It's a great question. Good eye. Others? Please.
I said I have a lot.
Yeah.
There have been recent studies showing that alcohol can damage DNA.
Right.
And given your research, I'm curious how that's impacted your personal view of alcohol.
Oh, wow. That's a really interesting question. That feels like a loaded question. No pun intended. All right. So we're now stepping into my opinion, right? This is just me. I would say it's interesting, right? And we talk about this actually in my biology of aging class. Every other day you hear something new in the news, right? So for example, I follow this pretty closely. Some days, right, you'll hear that a moderate amount of alcohol can actually be a good thing and it might allow you to live healthier and longer. Definitely too much alcohol bad, right? That's very clear. But I think it was just in the past few months, there was a study that came out suggesting, right, that any amount of alcohol is actually detrimental. My personal opinion, okay, is that these studies, it's not that the studies are wrong necessarily, it's that you're studying different populations of people and you're not always able to control for all of the variables. So some of the conclusions may be influenced by some variables that you don't know. I enjoy an occasional beer or glass of wine. Not everyone does. I will do it because it enhances my life, right? But I'm careful as well. And that's totally my personal opinion. We haven't used the fruit flies to actually test whether or not alcohol administration has anything to do with lifespan or damages DNA. I would guess it probably does. These are all good questions. Yeah.
Yeah, I've heard recently that less is better. The term occasional has flexibility in it. So,
right.
How about the Mediterranean diet? I've heard that's really good for us to focus on that.
Yeah, so he's asking about the Mediterranean diet, right? And interestingly enough, one of the student groups this semester, we just posted their video about a week ago, they studied the Mediterranean diet. Again, it's complicated because there's lots of things that go into the Mediterranean diet, right? And it's hard to isolate the diet from the genetics of the people who often consume the Mediterranean diet. But overall, it looks like there's a lot of positives to having a Mediterranean diet for many possible molecular reasons. I try to eat that as much as I can. It's also very tasty. So I like that. I think there's a question here and then up there.
The research, obviously, fruit flies, for all the reasons you mentioned, are much less expensive to get brains for, to learn something about as you trade up to bigger and bigger animals.
Yes, I will repeat
What is
it.
the trade-off? What is that? Can you do 5 times as many studies
All right.
as an mouse study?
So the question has to do with how much does it cost to do research with fruit flies compared to like a mouse, research with mice, for example. I purposefully, when I was a postdoc, chose to work in a fly lab because I wanted to work at an institution where I could do the research with undergraduates and I could do it if for some reason funding got difficult to get, which ironically enough is starting to happen, right? So yeah, I would say fruit fly research is probably less than half the cost of mouse research. It's up to you in terms of which you think, right, is going to be more applicable to human health, if indeed that's your goal. We learn a lot of things about the fruit flies that even if they're not necessarily going to be true in humans, they're just cool, right? I mean, there's a wonderful thing about studying biology and learning how different things in nature have solved different problems. I think the students love that regardless of whether or not it's going to cure cancer later on. And so that's why I chose to go with the fruit flies. It's a great system for working with students, and they're kind of cute. Question up top.
What's your opinion about eating smoked foods as far as your health goes? Chemistry shows that smoke contains many polynuclear cancerous compounds.
Yes.
Is this reflected against your longevity when you eat smoked fish, smoked meats, smoked foods, which are very popular in our culture? Is there any connection to longevity and eating smoked foods?
Okay. So a question has to do with smoked foods, right? There is definite scientific evidence that the chemicals that are created when you smoke foods or when you caramelize sugars, things like that, that those can damage DNA when DNA is exposed to it. So from that, you can conclude that yes, those could potentially be carcinogenic. Do they shorten lifespan? I don't know of anyone who's done a study on that, to be honest. And many people enjoy them. They're very tasty, right? So like many things in life, it's all about moderation. Our cells have evolved ways of dealing with that DNA damage. I actually teach that in my molecular biology class, right? Our cells are pretty good at that. But yeah, if you overwhelm them by eating it all the time, there could be problems. So it's all, in my opinion, it's all about moderation, to be honest. Are we done? I guess we're done. All right. Thank you, everyone. Thanks for your awesome questions. I hope you have a wonderful weekend back.
Thank you so much. Very much appreciated. And just a reminder that there is a full schedule of events for the weekend. And if this topic was of great interest to you, please come back tomorrow afternoon at 1:30. Shelley Payton, Professor and Department Chair of Biomedical Engineering, will be giving a talk, Breast Cancer and Beyond, Advances in Women's Health and Biomedical Engineering at Tufts. So enjoy the rest of your weekend. Thank you so much for being here. And thank you so much to Professor McVey. Thank you.